Welcome to this free webinar on RNA sequencing for infectious diseases. Today's session will introduce you to the RNA-seq data analysis and the upcoming online training program. This training will include methods of machine learning for bulk and single-cell gene expression studies, special focus on examples from infectious disease research, such as malaria and COVID-19 disease progression, immunology, and biomarkers. The upcoming OmicsLogic program, called OmicsLogic Transcriptomics, starting on April 16th, will be focused on how we can leverage RNA-seq to study infectious disease from the perspective of host responses, disease progression, and host pathogen interaction. One of the major focus will be on the application of bioinformatics to malaria-related research, where we will be looking at efficacy of vaccines and drugs associated with resistance and the complexity of host pathogen vector interaction. For this purpose, our team has already prepared a number of research data repositories and databases that will help participants identify high-impact data sets for analysis. We will also discuss various applications of genomics to the study of SARS-CoV-2, where selective pressure on various proteins will lead to the emergence of new variants. Such changes are already being seen as a result of increased immunity, vaccination, and other host pathogen dynamics. At Pine Biotech, our mission is to simplify bioinformatics for students, clinicians, and biologists, and we do this through our training programs known as OmicsLogic. The OmicsLogic bioinformatics training programs are running in 5 different regions with over 10,000 users all over the world. Our training is designed to allow participants to apply methods to curated and new data sets and develop independent projects that showcase what has been learned in the context of a personal portfolio of projects. As a result, anyone starting on this journey in bioinformatics can start developing a project of their own. In fact, we have already worked with many students who have completed this kind of training and developed interesting research projects that won awards and got published in peer-reviewed journals. These are some of those project examples. And to achieve this goal, we use a combination of online sessions like we're having today, self-guided study materials, which I will be introducing you to shortly, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. So in the upcoming 1-month period, you will have the opportunity to interact and collaborate with an international team of experts specialized in the areas of bioinformatics and data-driven research. Let me introduce you to the program mentors. We will have Dr. Harpreet Kaur, who is our OmicsLogic trainer and expert bioinformatician. She will mentor sessions and guide participants through the program. Her area of specialization is in cancer genomics. Dr. Mohit Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. He will also guide the project sessions and offer his expert advice on student projects. Ilya Brodsky is our CEO and co-founder. His experience in research and academia has led to many research labs and university collaboration and has been recognized for the simplified approach to omics analysis for education. Now, I would like to invite Ilya to talk more about transcriptomics for infectious diseases. Over to you, Ilya.
Thank you very much, Beepsa, and welcome everyone to this webinar. I'm going to just share my screen here. So as Beepsa mentioned, my name is Ilya Brodsky, and what I want to do in this part of this session is to introduce you to some of the topics that we will be covering in this program and share a little perspective on RNA-seq in the context of infectious disease studies using several examples in peer-reviewed journals that I find to be very relevant to what we have been studying over the course of the past several months with this program that is organized together with the University of Ghana, Noguchi Memorial Institute for Medical Research. So as already mentioned by Vipsa, this upcoming OmicsLogic program will be one of the components with a special focus on transcriptomic or RNA-seq data. In other words, we will learn how to analyze next-generation sequencing data to study gene mRNA expression. And that includes alternative splicing, variation, and gene set annotation. And so what we will do in the program is have quite a number of different online training sessions that I will kind of tell you a little bit more about, where we will both discuss, practice, and then apply various aspects of RNA-seq data analysis to different types of project data sets that we find to be relevant to infectious disease and immunology of infectious diseases. Specifically, we will review methods and computational environments where such large next-generation sequencing data can be processed, analyzed, and interpreted from a biological standpoint. And what's important is that we will touch on some aspects of RNA-seq data that turns this data into an excellent resource to understand the complexity of biological regulation, how it is affected by the immune response to pathogens, and to study these types of variations at both tissue and organ bulk RNA-seq to get a snapshot of what's going on, as well as a very detailed understanding of how differently this process can take place at the single cell RNA-seq level. And so, as you probably know already or can imagine, one of the major challenges associated with such big data is just the size of RNA-seq data. So RNA-seq data sets from a bulk RNA-seq experiment can be anywhere between 1 and 50 gigabytes, depending on the sample size. And single cell data sets can be even larger due to the large cell counts and also large sample size as a result. And so that's why what we do for all of those that register for this program is provide you with access to this T-BioInfo Big Data Analytical platform that was developed by the Tauber Bioinformatics Research Center at University of Haifa. And what essentially it provides you is a large disk with a lot of RAM where you can analyze these huge data sets online. But also I want to show you how the logic behind analysis is designed into the interface so that you can easily deal with large heterogeneous data sets from multi-omic studies. In other words, applying the understanding of the logic of analysis to a variety of different challenges. So the platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run various types of bioinformatic pipelines that include many open source scripts, packages, as well as proprietary analysis techniques relevant to different types of data. Different stages of analysis are performed on different sections of this multi-omics platform. And so just to show you how it works and why we use it in our training programs as well as research projects, let's go and check out how this platform works. But before we do that, let's make sure that everyone has access. So if you could go with me to this link, learn.omicslogic.com, you can register if you don't have an account yet. And if you register, you will actually get a free account that you can use on this platform. Okay, so if you could go with me to learn.omicslogic.com. And once you get here, you can click on login. Now, this is a free account, right? So this registration is free. You can sign up with social media, or you can sign up with your email address. And once you sign up, you will get a demo account on the T-BioInfo platform. So I want to show you, but if you want to follow along, you should create this account. And once you're in here, you will get an email notification. And in that email, you will find the details for the T-BioInfo platform. So as you are registering, I am going to head on over to the TBioInfo platform, and I'll wait just a couple more minutes for everybody to, whoever wants to, of course, to come to this link. So once you are registered, and you will see this in your email, you will get a link to the server TBioInfo platform. Now, what is it? It's a bioinformatics platform, a server online on the cloud that you can log in using the credentials that you will receive by email. And once you're in here, you will first just get a demo feel for what we are going to be doing. So I'll just guide you to see how this works. And then later on, I'll describe what are some of the sections that we're going to be using and covering in the training so that you both understand how to apply this, but also can apply it to a project of your own, a data set from the public domain. All right, so I see that a few people already registered. Great. So if you already have an account or you are logged in, you'll see your account right here. Maybe let me know in the chat if anyone wants me to kind of pause and wait for them to complete the registration. So if I don't see anything, I'm just going to continue. So next generation sequencing data, and you can kind of hover over this section right here to understand what it is, has revolutionized the way that we understand what happens at the subcellular level, because we can take a snapshot of all of the expressed mRNA, here speaking specifically about transcriptomic data analysis. And one of the first sections that you'll see right here is this section called transcriptomics, or RNA-seq chip parallel analysis of NGS and microarray data. And instead of going into this section, I'm going to click on this arrow going down. And here you'll see some demo pipelines. So some example pipelines that we'll just take a look to get a sense of what this is. Once you are here, you have this interface that is organizing various algorithms that you've probably heard about into a pipeline builder. So this is a pipeline builder that allows us to select various methods of RNA-seq data processing, error correction, mapping, construction of isoforms, as well as generating a table of expression, and then analyzing the differences between biological conditions. So let's just quickly build a pipeline so you'll see how it works. So you can click on Start. Every time you will see a pop-up that explains what is the logic behind the steps that we will be doing or that we have done up until now. So for example, here, you can already see an example of a pipeline that we'll be making. It starts from here, then goes into blue, red, green, and we end the pipeline. So here you have all of these colors explained. Blue is for pre-processing, red is for mapping, and then green is for quantification of expression levels. And so we'll see how this works when we build this pipeline. So after I select the first step, now I can continue on to select other steps that are highlighted in this dark gray. Following the example that I had from the Start button, I'm going to click on Trim-O-Matic, and you will see that one of the steps in processing the reads that come from the sequencer is to clean them from these adapter sequences and some bad reads that kind of degrade the quality of our data overall. So before we start analyzing, let's remove any technical sequences and variation that is not needed for our biological interpretation. I'm going to click on OK. Another artifact that is common to these sequencers is what's called PCR bridge amplification. You can see that step right here. What it essentially does is amplify the biological material so that we have enough of it to get a high quality read signal. However, PCR amplification, excuse me, does not work in a uniform manner and has some bias on some sequences. And we can detect that bias statistically by seeing an high coverage in very specific sequences. So we can eliminate this kind of bridge amplification using PCR by this additional script called PCR clean. After we have completed this kind of trimming, we can then proceed to select one of the mapping algorithms, and there's quite a variety of different mapping algorithms that might have a particular benefit for a study that you might find on public domain repositories of next generation sequencing data like NCBI. And so it's important to understand both the data that you're dealing with as well as the question that you are trying to ask. And the whole objective of doing what we are doing today is to illustrate that this basis for the selection of algorithms and building of your pipeline, the logic behind the pipelines is essentially what you will be getting most from this program. So if you're not familiar with why we are choosing a particular method and how this method ties into the overall objective of the analysis, this is why we will go through all of the methods here and kind of review some of the considerations behind why or why not to use this particular methodology. Okay, so we finally got to the step of generating an expression table. So this is a table of expression. And we have the option to perform differential gene expression, or we can just end it right here. Okay, so here you will already see that some of these are detailed, some of these methods are covered in our detailed online coursework that we will go through in the program that I'm speaking about. So here's the demo pipeline. Right away, you will find here some output files. So if you would like to just explore what is in these files, how did they get generated? What is the difference between different methods or different connections that we've made? Please go ahead and explore that right here. So again, for those that are just joining, the way we got into this section is by creating a free account on learn.omicslogic.com. And once you have created your account here, you will be able to find the details of access to this tBioInfo platform, as well as review some of the coursework that we will be going through in the context of this program. Okay, so as I mentioned, this is one of the challenges is big data. And so in this program, you will get access to these big data tools that will allow you to analyze RNA-seq data. And so first of all, we will prepare the processing pipelines to get the simple gene expression table or to perform differential gene expression, which are some standard analytical techniques typically used in the context of RNA-seq experiments. We will also look at ways to explore these big data sets that contain thousands of genes in tens or even hundreds of samples. And what's important is that they can reveal some important variation between biological conditions. Like, for example, in this study, you can see principal component analysis of 3 different conditions across 2 different types of viruses. A virus, this is from SARS-1, so a virus with ORF6 or a delta ORF6, a mutant that does not have that gene. And so one of the questions that the authors posed in the study is what would be the difference in disease progression over time in cell lines, and how can we develop a set of contrasts to start asking the question in the context of differential expression and ultimately for biological interpretation. As you can see in that example, it is not a simple study where you have a condition and a control or 2 different conditions that are exactly identical. In fact, what you might suspect is that there are several factors at play. Maybe it is the type of media that you are growing your cell lines in. Maybe it is the differences between pathogens. Maybe it's some kind of a manufactured condition like a delta mutant virus or something like that. And so what we will go through is not just exploratory data analysis and differential expression, but also we'll look at some of the ways we can apply regression to understand what are some of the relationships and important factors that contribute to observed effect in our data. For example, we'll talk about factor regression analysis and what unsuspected results it could shed light on when we approach this data from a data-driven perspective. Ultimately, though, we will talk about gene set and pathway enrichment. And so we'll talk about how some of these analytical methods can provide us with important values to differentiate between different gene sets that could be annotated. Thankfully, we have a lot of annotated resources, especially for human and mouse animal models. And so we can start using some of these references to really understand the biology behind what's going on. So we'll talk about some of the things that go into biological interpretation. So as I mentioned, this program, RNA-seq data analysis training program, will start on April 16th. And what's important about this is that we will both explore some of the relevant tools kind of from a theoretical perspective. We'll review some literature that explains when and when not to use some of the tools and what are some of the considerations when you're building up your pipeline. But we'll also talk about how to apply these tools to study infectious disease manifestation in different types of hosts to understand the intricacies of disease progression and how we can study the immune responses and the manifestation of clinical phenotypes that we typically associate with disease. So I think it will be an exciting program, and I'm hoping that all of you will be able to join to participate in the sessions for the training or to design and develop your independent project. And I'm sure that Bipsa will continue speaking about this at the end of today's session. But before I pass it back to her, I want to briefly review some of the sessions that we will have for this program. So Bipsa, if you could please put the link for the training program schedule. So if anyone is interested to kind of get a snapshot, an overview of the different sessions, what topics will be covered, as well as what are some of the themes that we'll touch on. It is going to be on this link, transcriptomics.omicslogic.com. Thank you, Bipsa. And so you can see here a detailed overview with some of the dates when these sessions will take place. They will be interactive sessions like we are having now, where you will be able to ask questions and practice some of the things that we do together. So let's review some of those sessions. What do we have planned? Let's go through the slide. All right, so it will start on April 16th, and we are posting here the times in GMT time zone because we are working together with the University of Ghana, where many of the participants are already a part of this overall training on bioinformatics for infectious diseases, and hopefully this will be another asset in their toolkit to study variable immune responses. So we'll start off by talking about processing transcriptomic data. So we'll talk about how is this data generated, what is in this data, what are we looking for, what is the logic behind what we can do with this data, and what are some of the steps that we could consider depending on what we are trying to accomplish in our analysis overall. We'll talk about some of the outputs and how these outputs could be explored. And we'll see how they are representative of the raw data. So we'll actually take a look at what the raw data can reveal about these observed recorded values that we obtain in a structured format. We'll then kind of turn to a session on quantification, normalization, and visualization. And here you will get introduced to some of the tools in R or Python that you could use to quickly explore using different aspects of summary statistics and log normal transformation to understand variability and how that variability is representative of the design behind the data set that you observe, right? So somebody actually took this data and prepared that data. And so one of the things that we want to understand is how can we explore the variation that is unwanted, technical noise, as opposed to how this data set could be prepared for downstream analysis. So one of the things that we will use in this session are several practical coding exercises to help us load data in R and Python, explore this data using visualization tools, map this data onto different types of categorical values from our sample design. We will look at different types of applications when we have data that is already processed or data that is raw, and how to deal with that. So some of that coding experience is something that you can already go ahead and try on learn.omicslogic.com. Then we will turn to differential gene expression. So we'll take a look at these popular packages called DSeq2 and EdgeR. And there we will learn both about the logic as well as the differences between these different methods and how we can use the different values in these methods to really select the genes that are helpful for biological interpretation. In other words, which ones are actually different when we talk about thousands or even hundreds of thousands, at least tens of thousands of genes, and how do we select the ones that are most interesting. We'll also look at some examples to understand what is some of the variation that we should expect in our data and how to differentiate between statistically significant and insignificant variation for our downstream interpretation. As I already mentioned, we will not only touch on some of the basic statistical analysis, but we'll also talk about regression analysis. That is a powerful tool to understand variation that is not immediately obvious and understand how we can extract meaningful insights from complex data sets. Biological interpretation is also an art that you master as you apply it to a variety of data sets. And so we'll talk about different ways to annotate, visualize, and associate gene expression data with biological processes. And so we'll look at data sets that contain specific what's called enrichment of gene sets. And one of the tools that we'll talk about is what's called gene set enrichment analysis. We'll talk about what are some of the methods and settings for this analysis, and what are some of the outputs, how to interpret these outputs, and what we can learn about the biological processes and differences in different data sets that explain this type of variation. Now, as you know, gene expression is continuous data. And because it's continuous data, there are many tools for unsupervised and supervised machine learning methods that are going to be useful to both explore the data that we have and understand how this data represents some of the major trends in the data that could explain biology or differences between different types of samples that we study or different groups of samples that we study. So we'll take a look at how do we apply different clustering techniques and how do we use different dimensionality reduction techniques to understand such variation. We'll also talk about supervised machine learning or classification. We'll take a look at some of the commonly used methods that could help us deal with large data sets when we have very specific and maybe not often statistically significant patterns that we cannot find in using normal multivariate analysis methods. So we'll talk about random forests and how we can perform bootstrapping and bagging. How do we use that to both identify, train, but also how to find specific features that are important for separation between classes. So we'll look at feature selection. Now, what's important in this is that many programs actually teach dimensionality reduction and feature selection from a computer science or a statistics kind of point of view, where what's important are different metrics that allow us to actually perform the selection. But I think what is important for us here in the context of the biology behind these features is to really understand what are the specific things that we could be looking for to interpret the feature selection process, and how do we combine our biological understanding of data sets with some of these statistical measurements that we could then use to automate the process. Now, what's important today in any kind of RNA-seq discussion is to talk about single-cell RNA-seq. And so what we'll do is we'll talk about the differences between bulk and single-cell RNA-seq. It provides us with a very clear picture of what's happening at the single-cell level, but comes with a variety of challenges that we need to understand. Those challenges have to be addressed at the level of processing the data, so preparing it for analysis, as well as with the downstream interpretation of these results, because as you know, these are zero inflated data sets. So they have a lot of zeros. And so we're going to be talking about how do we actually effectively use some of these annotation techniques to find UMI or UMIs, and how we can then prepare a meaningful picture of what is in our single cell data sets. Now, throughout this program, we will refer to projects and case study data sets. So we will not be just using these methods just to understand kind of the practical technical aspects of these methods, but we'll look at examples where these methods have been applied effectively. And so we'll take a look at some of the research methods included in publications that reveal how these types of methods are used to understand infection by Ebola virus, by SARS-1 and SARS-2 viruses, as well as malaria. And what you'll find a lot in this context of the infection is that we can compare infection as a result of similar viruses. So for example, we will talk about how other respiratory viruses that also cause severe respiratory disease can compare to SARS-CoV-2. We'll talk about the immunology, what's called the cytokine storm, both in SARS-CoV-2, but also we'll talk about Ebola, which, as you know, is one of the pandemics that was the start of this massive scale next generation sequencing effort. So we'll learn some about Ebola there as well. But we'll also talk about how such transcriptomic data is being utilized in the context of clinical and biomedical research, in the context of epidemiology, but also in the context of drug development. So here you can see some examples of how network analysis and enrichment analysis can help us understand the different types of responses and targets, ultimately as nodes in those networks that can help identify compounds, protein-protein interactions, and various transmission prevention techniques so that we can understand the molecular drivers or components that help explain some of those considerations. So after you complete this program, you will have the opportunity to speak about the transcriptomic methods. When you read papers, you'll understand those methods. When you explain your own analysis, you'll be confident in knowing how to explain why you've chosen a particular method or what did this method do. And you'll be able to share an example project, either from one of the projects that we provide or a project of your own, so that you can have an experience of applying bioinformatics techniques to a data set and kind of going through the full process from the very beginning of identifying that data set to then processing the data set, preparing it for analysis, and then finally communicating the story, the biological story behind what you did, and getting feedback on that from the other participants of the program, as well as from the experts on our team. So I hope that all of this answers a lot of the questions that you've had. If you still have some questions, please do post them in chat and I'll be happy to answer those. And I will pass it back on to Bipsa to discuss some of the technical aspects of registration, cost, and the details of the different resources that you will get access to. Bipsa, over to you.
Thank you, Ilya. So for different parts of the session, we will be leveraging the OmicsLogic resources. To learn about transcriptomics, this will include the asynchronous coursework that you can find on learn.omicslogic. So
sorry. So I do see a couple of questions. So let me just briefly address. I see one question. So one of the questions here is, how will this program help in getting employment? So I just want to briefly mention that, we are not advertising some kind of a, career change program, but in general, if I can briefly speak about the trends in the amount of data that is being generated by research centers and labs, as well as in the industry in both clinical and pharmaceutical and biotechnology industries, you will see that there's a growing demand for biologists with computational skills, as well as data scientists that understand the biology behind the data. So this is such an important trend that NIH, the National Institute of Health in the US, has started a specific program with a focus on data science to help train up both biologists and computational engineers to understand bioinformatics. And I think this is evident also if you search anywhere online. People with bioinformatics degrees or bioinformatics capabilities are in high demand. In fact, salaries for biologists, wet lab biologists, as opposed to bioinformaticians, also tell the same story. So you can get 1.5 to 2 times as much as a data scientist or a bioinformatician as opposed to a wet lab biologist. And so what's important here, I think, and what the employers are really looking for isn't necessarily just a degree, because they understand that many students can go and, based on this kind of obvious trend, go for a degree and get some basic training. I think what employers are looking for is your ability to think critically about the data with an understanding of the methodology, and your ability to apply this methodology in a correct way to a real biological challenge. And that is not a trivial thing to demonstrate, but I think one of the best ways that you can demonstrate that is by showcasing a project or even multiple projects that you have been able to conduct with the explanation of how you've used specific tools. And so, I'm not going to sell this as a quick fix. You want a job as a bioinformatician, this is a program for you. That's probably not the answer. But I would say that this journey into a career as a bioinformatician starts from this kind of an experience where you can take a data set and analyze it, working with experts and other collaborators to get a real impactful kind of a project out. So that's my brief answer to that question. So I hope I've answered that based on the chat here. Thank you, Bipsa. Go ahead.
Thank you so much, Ilya, for answering that question. So as I was again mentioning to everybody, so Ilya has explained very well what are the different resources that we have. Now, again, to explain that once you are creating and logging to this platform, so let us start here together. What this will give you, the free level login, is also a demo account. So as you see, I'm already using my email ID. You can also utilize any of your social logins. So once you create an account, you will also be sent an email from dev@pine.bio to create account, set up your login details for the server tbio info platform. So at this free level, you can start exploring some of the free lessons that are available. And let me take you to the transcriptomics course. So if you notice here under the transcriptomics course, there are several different lessons. So if you keep on loading more, you will have different topics that are covered in all of these lessons here. The first three of these lessons, as you see, are free lessons. Any of the lessons marked with a star are the lessons that require subscription. Anybody who is already registered for the program will have access to all of the lessons which are mentioned here. They do not have to take up subscription otherwise. But if you're interested, you can go ahead and also take subscription to just these lessons, and you can reach out to me for those links. But similarly, not just for transcriptomics, we also have other courses that the free level on learn.OmicsLogic will give you access to. So for example, somebody who was interested in microbiome data, right? Or learning more about microbiome. So here are some of those metagenomic lessons for you that you can go through, the introduction to metagenomics that will help you get started in learning about what metagenomics is and what is, how to understand this kind of data. And again, you will notice some lessons marked with a star. So in this way, we have designed this page so that at the free level, you are able to get an understanding of what bioinformatics is or what are these different types of omics data sets. So if you are somebody who is just getting started with bioinformatics, we definitely recommend you the Introduction to Bioinformatics course. This is a completely free course and will help you have an understanding of how bioinformatics is useful in different domains like healthcare or research, as well as even in military and other areas. So please feel free to take this course, and you can explore all these different projects and courses right here. So you'll find the courses around here, and each of the participants would be identified by their email ID. So as you see, here is my email ID. And if I go under here, I would be able to see what are my achievements so far, how many points I've earned, what is my activity on a daily level. So we can see all of these from the Learn OmicsLogic platform. And what I wanted to also let everybody know is that how to register for this entire training. So this is the link that we have for the training program. Now, to get started, we have already seen that in this page, you will have all the different dates. So we are starting this Friday, which is April 16th, with the first session of this entire 1 month program. And we will end with single cell RNA-seq on May 20th. Now, let me explain how you can register for the program. So if you come to this page, you can either click on pricing or on get started, and you will be given these three options to register for the program. And I will also explain what these three options mean. The beginner license gives you access to all of these subscription courses, which we have shown here, all the courses marked with a star, as well as educational access to the TBioInfo platform, and also the group session. So you will be able to take advantage of the full program, start working on a research project or a research proposal, because a project may not be feasible in a short period of time in this 1 month program. The intermediate license comes with mentor support, which means that if you are somebody who thinks that you want to work with a mentor, in addition to the group sessions, we recommend the mentor support. This will come with a one-on-one session with your mentor, and he or she will be able to answer any questions besides the group sessions that you may have, while the advanced license gives you an additional 30 days. So as you see, the program ends on May 20th, but for anybody who has an advanced license, the program will end on June 20th. You will have an additional 30 days to not only have a project proposal, but also possibly work on your project and try to complete it or make it presentable so that you can do a poster out of it or present it at conferences, and or work in your project and make sure that you can take it to the next step. So these are the three licenses that are available as part of the OmicsLogic program, the OmicsLogic Transcriptomics program. So please feel free to explore the licenses, and if you have again any question, here is our email ID. So you will be able to reach out to me anytime you have any question. Now, let me go back and talk a little more about some of the program structures and objectives. So what we will learn during this entire 1 month training is about the concepts within the immunology of infection and disease progression, which will include some topics like central dogma of molecular biology, immunology of infection, molecular disease markers and stages, host pathogen interaction, response heterogeneity, gene regulation, and target pathways. So we will also discuss several bioinformatics methods such as processing of NGS data, variant calling, quality and processing of RNA-seq pipelines, statistical analysis, machine learning methods such as clustering, principal component analysis, classification, as well as annotation. And once you have already become familiar with these methods, we will give you the option to first go through example projects. So Ilya has already explained to you how we'll be utilizing these example projects and help you understand how to work with these kinds of data sets, and then slowly giving you the opportunity to work on your own projects as well. So once you complete this training, you will be able to understand how to do the following, which is read and understand bioinformatics literature, perform basic NGS data analysis processing steps, analyze data and form hypothesis, prepare data for machine learning, cross-check signals with gene databases, know what type of data is required to answer scientific questions, create your own bioinformatics analysis, and present your results in an organized way. All of these skills, which are very, very essential in bioinformatics and can also be translated to other fields. And after achieving the learning outcomes, you would have learned some of the major best practices in the field of bioinformatics. You will be exposed to R and Python, the two very powerful programming languages. Bioinformaticians will need to understand how to use t-tests, regression, and other tests, analyzing and management of data in order to communicate important information, which is also another best practice in bioinformatics. We'll also touch upon pipeline development and utilization, and ultimately completing the program will help you enter the field of bioinformatics, since you will learn several of these best practices in bioinformatics. And once someone completes the program, this 1-month training program, they would be given a certificate of excellence or certificate of completion based upon their performance during this entire 1 month of training. So yeah, we offer certificates whenever someone completes a training program. And you already know that if you are interested in joining the program, again, the link is here in the chat box that will take you to the registration page. So please feel free to complete your registration here. And we also have these few groups on LinkedIn and also in Facebook. So let me post the link again here in the chat box so that anybody who is interested in joining these groups will be able to go ahead and join. And you can also share with us any feedback that you have about the program and the resources, and start taking part in some of the community discussions that we have in our groups on social media. We are also active on Instagram. You can find us at Bioinformatics Spine, where we keep on sharing interesting images, interesting topics, interesting research articles, as well as featuring some of our bioinformatics community members. So here is the Bioinformatics Spine Instagram handle if you're interested in going there and asking us any questions, or if you're interacting with us there. So please feel free to reach out to us with any questions that you may have even after this session. And I want to take a minute here to ask everybody, do you have any questions or would like to ask us any questions? Please feel free to post them again in the chat box, or to write to me at marketing@pine.bio, and I can help you address some of those questions. And again, I want to thank everybody for joining today's webinar. I hope that this was informative for everybody, and we look forward to seeing you in the upcoming program. Thank you all.
Thank you, everyone.